Hello.
Okay, it says I'm live on my side.
Okay, so let's get a confirmation from the audience that...
But I can't hear anything.
Okay. Okay, it's clear for the audience.
Okay, I don't see you, unfortunately. It says stream is unable to connect on your side.
Okay, so
you can introduce me, but I won't hear you, unfortunately.
Okay, go ahead and share your screen. So this is a strange situation. Dr. Fairhall is unable to see or hear me.
All I have to go on is the chat. So maybe just tell me what to do by chat.
Okay, hover over your...
Share my screen.
Yeah.
How do I do that? How do I share screen? Yeah. I see.
Okay. Your screen is visible.
I'm now sharing my screen. All right, let me get that up. Okay, now I can't see any any text either, so I'm just, can I unshare again?
Okay.
I'm just going to assume that you're all there, and then you can see me and you can see the screen, and someone should just, Guillaume should call me if that's not true. So yeah, thank you so much for the invitation to be here. It's really an honor to join you for this symposium. Thank you so much to Surya and the organizing committee. And also thank you to Unique. You know, as you might have been told earlier, both Konrad and I joined as part of the advisory team for Unique this year, and we're both really thrilled to be able to be part of this amazing organization. Also, congratulations to the speakers from earlier. You all did an amazing job, and it was incredible to be able to see so many people join from all over the world. Okay, so I'm going to pick up on some themes that you've already heard quite a bit about, including in the last hour, the relationship between learning and the hope of being able to understand learning from an algorithmic point of view, and how that's done in biological systems, and hopefully something, what we can learn from biology in order to potentially update our text. Oh, my slides are not up. I did share my screen. No, I don't know how to not. Okay, let's see. How do I do it? I can try again. Yeah, thank you very much for, this is very cool. I don't know what's going on here. No, I don't see my video either. Maybe we should
disconnect,
reboot, because I think something horrible has happened. It's really awesome. But in fact, I'm going to take a picture of
it. Okay.
Why don't you drop me out and bring me back in again?
Okay, so apologies. Let me just try to disconnect Dr. Fairhall. I'm just inviting her.
Hey there.
Adrienne.
Still can't hear anyone. So what I'm going to do is try sharing one more time. And if that doesn't work, my backup will be to try this on my laptop instead of on this computer. So let's just see if this works. And you're going to have to tell me by text if you can see it or not.
Sure. Okay.
Great. Yeah, okay, that looks better. Is that possible that that is better? Yes, all right. Okay, let's go from there. Fortunately, I hadn't actually changed any slides as in what I was saying before, and we'll just head on. Right, so where I started was the actual title that I gave you guys for the talk. Where I am going to take it is to dive into a particular system, which is the birdsong circuit. That's a system I've been working on in sort of dribs and drabs for the last few years, and increasingly have a number of projects in this domain. And I would like to tell you about it because I think it's a great example of where sort of biology and learning algorithms really, really meet. So let's just start by kind of the big picture issue, which is lifelong skill learning, right? Motor learning is a great example of a skill, right, where you can quantify exactly how well you're doing. We would like to understand how brains learn to produce precise motor behaviors. One important part of that is the ability to be variable, right, to generate variability, to learn from the variations that you make. However, you're not always practicing, right? Sometimes you want to do that action as well as you can. And so we also need to be able to dynamically modulate the amount of variability that we inject into our movement in order to be able to operate in these different contexts. And we also need to be able to maintain skills throughout our life. So this is my son, actually, who's no longer that small. And he took up tennis as a kid, and it was sort of fun to see him learn those skills. But that also required, if I toggle to presentation mode, I think I am in presentation mode on my side. Is that not looking like presenting there still? Maybe there's a delay in this. I'm going to wait. Slides are not changing. This is a tough forum. Still not changing. I vote Zoom next time. How about that? Still not changing. So I go out again. Screen's frozen. Okay, let's try again. I'll share one more time. I'm now, I went out of presentation mode. I'm going into presentation mode again. Does that look like presentation mode to you? I think it'd be much easier to call you. Hi there. So, yeah, what do we do? No, I don't. It looks like it would work better without the presentation mode. Had to share screen. share two screens. Huh. Let's see if I can close video. Now, is that stopped sharing now? Export it as a PDF. Oh, I hate USB. So when I go, let me tell you what happens when I try my share screen. I get window or screen to share, select window or screen. And then I can go to Keynote, or I can go to, that's interesting, right? So there's either Keynote 3 or Mila, or the name of the talk. So I'm going to, I think I've tried both, but let me try this one and see if that's better. That's what I want. Now I'm running the talk. Did that work? So, does that look like a proper slide now? Like there? Did that change? No, I don't see that myself. Okay, I just see my feed, because when I go into present mode. Hang on a second. Okay, where is my video now? I've lost myself now, so sorry, I cannot find where I am anymore. So I think I closed my own window of my moving other. Okay, let's try this share screen. Now, so tell me again. So window or screen to share. Select window or screen. I could do Keynote 3, or I could entire screen. Is that what we want? Okay. Sorry about that. All right. Now you should see yourself, Surya. Do you? Sorry, everybody. And now, how's that? All right. Okay. I am so sorry. Good. Let's take it from there. Preamble, blah, blah, blah. Okay, bye.
Thanks, Guillaume.
Oh. All right, where we were. So I was telling you about lifelong skill learning, that one would like to understand how one learns to produce precise motor behaviors. And that involves practice and performance. It involves being able to modulate the amount of variability. And I showed, not you, but myself, this picture of my son, and was going to tell you that when he took up tennis, then that sort of forced me back on the court. And I only learned when I was about his age. And so one thing that one discovers when one tries to regain a skill as an adult is that you still have it. And even although we're well beyond our most plastic period, we're able to maintain skills and adjust to the physical changes of growing up and getting older, despite all of the downsides, I would say, or physical downsides of that process. And one can look at the superstars like Serena Williams, who's sort of back on the court even after having a kid. All right, so why have many people thought about birdsong as a great model for motor learning? There's a lot of great reasons. Are you able to, oh, no, you're not able to see my mouse with this display. So there, it has, for one, it's a highly stereotyped behavior. In particular, the typical bird model that many people use, which is the zebra finch, has essentially one song that it repeats over and over again. That is great for doing any kind of quantitation because we know exactly more or less what the target is, and we can characterize variability and learning very well in such a very limited situation. So it's like a kind of sandbox for motor learning. There's been a lot of work done on the bird brain, so it has very well characterized neuroanatomy, the different components of the specialized circuitry that the bird has for learning have been identified, and their roles more or less mapped out. Birds are also trial and error learners. That's a very, very important part. They don't just get born being able to sing their song. They have to learn how to do it. And so that gives us the opportunity to see how that process works, how the process of variation and learning over many, many trials can work. Birds also retain some plasticity throughout their life. So once their song is learned, even as adults, they're able to make some slight variations in the song, and that so adds an extra level of interest to the problem, because now we can also work to understand how the song is maintained and how that additional small component of learning might be realized. What's also, I think, very fun about studying a model like zebrafinch is it has a very simple song, but there are some variations. It's able to generate sequences of syllables where sometimes there are sort of extra syllables added. And so it's the very beginning of sort of a more complicated process that starts to go toward something like language learning. And as one moves from zebrafinch to other kinds of birds, those birds, can generate much more complicated songs. If you look at, say, canaries, they're able to generate long sequences of different syllables. And so the understanding that we can build of song learning in the zebrafinch, we can kind of extend out sort of step by step to thinking about how much more complicated and variable and maybe more grammatical sequences can be biologically implemented. So the goal with the work here is that we hope to connect learning algorithms as defined, at the algorithmic level with the biological substrate, given all the advantages of the zebrafinch circuit. And a lot of people have looked at this particular problem from a theoretical point of view. Some of them here. All right, so there are two phases to the manner in which birds learn song. When they hatch, they initially don't sing at all. They spend quite a long time just silently listening to a tutor, typically their father, who will sing over and over to them. They listen to that song. They develop, presumably, an internal template before they start practicing the song themselves. And when they start singing, they generate a kind of babble that doesn't look much like a song at all. And then over tens of thousands, of trials, that song eventually morphs closer toward something like a copy of their father's song. Every animal will induce slight variations from the tutor to their own individual song. But once they've reached their personal song, that song kind of crystallizes and remains fixed for life. So here's how that looks. So this is still during the learning phase. Every one of those dots represents the distance from the bird's produced song to a final target song. And you can see that trial by trial, so every one of those dots is a song. They sing many, many songs in a day, and there's a lot of variability in the songs that they produce. But over this period of days leading up to song crystallization, they are kind of converging toward a better, the final version of their song. All right, so birds, as I said, can also maintain the ability to learn even in their adulthood, even after the song was crystallized, and that was really beautifully demonstrated by Anderman and Fee, so Michael Fee's lab. So what they did was to select a certain region of the bird's song. So you see here in panel B, the targeted region, where there was approximately, nice, clean harmonic stack. And they would analyze the frequency of that harmonic stack and then set sort of a target where they wanted the bird to move that harmonic to, right? So that's what you're seeing in the panel below, that spectrogram is some threshold such that, they were trying to kind of train the bird to change the frequency of that syllable to be higher. And the way they would do that was that when the bird would sing a syllable, at chance in the range that's in that purple regime, they would do nothing. But if it sung the syllable with a frequency that was in the other regime, they would give negative feedback. And they would do that by, on online processing with this DSP, by giving a kind of, a sort of aversive sound that was played by this microphone right against the bird's skull. So it would get this sort of almost integrated into its own song. It would get this sort of unpleasant feedback sound. And so by doing that, what you can see from panel D is that they're able to kind of push the syllable around, right? So every day they would sort of set a new threshold and get the bird to shift the pitch of that syllable day by day. And they could kind of keep moving it around indefinitely. Right, so let's do a little crash course on bird song system anatomy, right? So here's a very simplified picture of the nuclei that are involved in birdsong learning. And we're going to be coming back to all of these, and I've tried to color code them to sort of help you out, remember who's who. So the song production piece, right, the motor circuitry is driven by HVC. We'll see firing patterns from HVC later, but HVC approximately acts like a metronome to the song. It kind of lays down the temporal pattern of the song. That HVC drives RA, which is a motor area that then drives the neurons that drive the muscles in the syrinx. And that is the key part of the song circuit that's required for production of the song. But there's this other piece, what's called the anterior forebrain pathway, all the regions in blue, LMAN, area X, and DLM, that are required for the bird to be able to learn its song. So if those regions are ablated, then the bird never learns a proper song. And also, we've drawn in here, because we're going to come back to this quite often, area VTA, which is like mammalian VTA, which, as many of you will know, is responsible, we roughly believe, for monitoring online error and giving dopaminergic feedback to assist in learning. All right, so that is the picture in the brain. We're going to simplify it down to this cartoon. You're going to see this cartoon quite a lot. So to re-summarize, the pieces we're going to be looking at is this cortical motor pathway responsible for song production, anterior forebrain pathway, the three nuclei. Here, area X is analogous to the basal ganglia. Area DLM is analogous to thalamus, and LMAN is a cortical-like area. So what you'll see from this simplified architecture is that HVC both drives RA, and it also drives this anterior forebrain pathway. And all the action of the anterior forebrain pathway, all the role it can have in learning, is through this output from LMAN that projects into RA. All right, so many years ago, Terry Sanowski and Kenji Doya sort of pointed out that this circuit could be a very nice model to think about reinforcement learning. That is to say, one could think about the HVC to RA pathway as kind of the actor. And they speculated that the role of the anterior forebrain pathway was kind of to act as an experimenter, to inject variation into the circuit such that HVC would learn properly how to wire to RA so as to strengthen advantageous fluctuations that led to a better outcome. So what we're going to go through today, I have a number of projects working with a bunch of different groups on birdsong, on different components of this circuitry. Part of it, we think about models of learning. Part of it, we think about how the anterior forebrain pathway works to generate and to modulate the variability that drives song. I also have a project working on VTA, examining the information content of the dopaminergic feedback, and another project with Carlos Lois's lab thinking about HVC, how it generates robustly sequence generation and it maintains that. But we're really going to focus on these two pieces, these models of learning and the origins of variability. And the very first place we'll start is with models of learning. So let's go back a little bit in history. First, let's have a look at what those firing patterns look like. As I mentioned to you, HVC acts kind of like the clock of the song. So individual neurons in HVC fire very sparsely. They fire once throughout the song. And they fire in sort of a sequence such that different neurons firing will sort of tile the entire time course of the song. Now remember that HVC neurons are driving RA neurons, which are driving muscles. Given muscles have to participate multiple times, there's only a limited number of them, I think 13 muscles in the syrinx. And so any given muscle is going to have to participate in multiple kinds of syllables. And so, as one would imagine, if you look at an individual RA neuron, that is going to fire, multiple times throughout the song as it helps to shape the nature of syllables at different times throughout the song. So the learning problem is, how does HVC, right, a neuron firing at a particular time, wire up to drive the RA neurons? How does it know which strings to pull, right, on the muscles in order to drive the song appropriately? And so we're really just going to focus on these three pieces: HVC, RA, and the role of potential role of LMAN. So this is a kind of classic model that Eilif developed when she was a postdoc with Sebastian Sohn. So we're just thinking again about the connections between HVC and RA. What they proposed was that LMAN can act as sort of an instantaneous generator of fluctuation, on a very rapid time course. So the earlier work by Doya and Sadowski had just postulated that on every trial, on every song, HVC might add, a constant, and that constant would affect the quality of the song. Instead, in this case, they were really thinking about those fluctuations as acting on an instantaneous timescale. And so HVC generates, essentially in their picture, just a Poisson random spike train that is added into the RA neurons. And then if those fluctuations co-occur with an input from HVC that acts in a way to improve the song, then this critic is standing by, ready to increase the strength of the synapses from HVC to RA, right? So it has access to the information about the inputs from HVC and from LMAN. It knows whether or not that fluctuation caused a favorable change in the song, and it will act to strengthen those synapses. So zooming in on those synapses, they're receiving inputs from HVC. These are plastic. There's this empiric synapse from LMAN, which is injecting random noise. If that noise acts to improve the song, then you'll strengthen the input from HVC. So we wanted to take that model and use it to model an effect that one of our collaborators, Tim Gardner, had seen experimentally. So in all the models of birdsong that have been made, one has taken that picture of HVC very seriously, that it acts as a scaffold to the song. It fires very reliably and consistently at every time throughout the song. What Tim saw, Tim was able to do was, he was one of the first to use calcium imaging in the bird brain. And so opened up the skull of the bird and was able to record over very long periods of time. What you're seeing here is just the song itself at day 1 and 405 days later. What you'll notice is that the behavior is extremely stable. The bird sings a very, very consistent song over very long periods of time. We had always assumed that that was due to extremely stable firing in HVC that acts as the scaffold of the song. What they found is, when they observed these neurons over very long periods of time, a small fraction of them in fact changed their firing. And so you're seeing here an example of a neuron that's firing very consistently at a particular time and starts to fire at a different time later in the song. And so they saw that overnight, sometimes some neurons would drop out, just a very small fraction of them. And so we wondered what impact that would have on the song model. So what we did was to implement the same model that Eilif had implemented, but we added to it this minor perturbation that from night to night, some neurons from HVC, a very small fraction of them, maybe 5%, would stop firing. They would just sort of skip firing or stop firing altogether. And then we asked, what effect does that perturbation have on the learning in the network? And so here you're looking at how the error behaves as a function of time over trial to trial, right? So every point here is in this trace is the error between target song and the performed song. So in our model, when we approximated the performed song just through 2 motor outputs. So they each had a target waveform that had to sum together to generate the target song. And this is the error between the trained network. So without any perturbation, you get down to a stable error of around 1,000 and whatever units, and it just stays there. When you add perturbations, that's what you're seeing in the black trace, that after the addition of a perturbation, or when some of these neurons shift or drop out, then the error of course increases. But over the course of time, that error will return back. So at this rate of perturbation, you can see that the error doesn't actually get back down to the original error. There's always a bit of an increase. So you do take a small hit in accuracy adding this kind of perturbation, and you can see that from the error improvement, right? So the more perturbations you do, kind of the worse you get at about a 10% level. But that change, or that new network that was trained under perturbation, did have a couple of advantages over the unperturbed network. And one was, what if you had to learn to change your song a little bit, right? So in the case of the experiments that I showed you from Michael Fee's lab, when they forced the bird to sing at a different pitch, we mimicked that by taking our original desired output from our two motor outputs and adding to them a perturbation. One could also imagine that this might happen if, say, you had injury or damage to a muscle and you have to learn to fire it differently, or you grew. Right? So what we saw was that the networks that were trained with perturbations adapted to these perturbations in the target song more rapidly. So now you're looking at the time course of the relearning error, and the different colors, different traces, are networks that are trained under different numbers of perturbations, ranging from no perturbation at 0 to 50 perturbations, which is the yellow one. And the more frequently you add in that perturbation, the more rapidly the network learns to retrain to the new song. So that relearning error is more rapidly learned, dealt with when you have a network that is trained under perturbation. We can also ask about the robustness of the network to, say, damage in the motor output. So here we ablated neurons in RA and asked how much of a problem was that for the network? How badly did the network perform in the presence of perturbations like that, or ablations? And that's what you see here. Again, as a function of the number of perturbations, the more you perturb the network, the less error it suffers under ablation of RA. So it's not just more adaptable in that it learns more rapidly to adjust to perturbation, but it's just intrinsically more robust to damage. So what's the reason for that? What's happened in the network to make that true? So if we go back in and we just look at the weights that connect HVC to RA, here we start with random weights. We train the network with no perturbation. Is the top plot. If we're doing this, 50 perturbations is the bottom plot, and you can immediately see by eye that they have quite a different structure, right? So the weight matrix that's trained without perturbation ends up having these significant correlations, temporal correlations between neurons that encode nearby locations in time. Right, so we can look at how strong that effect is. We can just calculate the degree of correlation in the weights as one, the weights in between correlating the HVC to RA connections as we move along the HVC chain, and where that index is what here is delta t, so the difference in time that the HVC neuron fires. And indeed, you see that neurons that are trained without perturbation are strongly correlated if they fire at neighboring times. And the reason for that turns out to be that they well, I'll get to that in a moment. So just to show you a bit more of that, so perturbations break that degree of weight correlation, right? So if you look at the strength of correlation and how it increases over time without perturbation, you add perturbations and you can see you're bringing that correlation way down until ultimately with 50 perturbations with very rapid rate of perturbation, then the correlation is very small. Why that is that we were using relatively realistic inputs from Elman. So Elman we took it to give random perturbations that were Poisson distributed, but when they impact the HVC to RA synapse, they are, biology tells us that those synapses are NMDA synapses, which have quite long time constants, to the order of 50 or 100 milliseconds, which means that neighboring neurons or neighboring HVC inputs are going to be subject to perturbations that are correlated up to a few hundred milliseconds. And so we believe that that is the effect that's generating the correlations in the weight matrix. But then if we perturb, we break up those correlations and we distribute the drive for some features among more HVC neurons that are more widely distributed. So another way to look at that is that as we train the weight matrix, it very rapidly comes down to a very low error, but then slowly this degree of correlation builds up, right? So that if we think about just plotting two weights in the space of two weights, the solution finds this rapid solution, but then will gradually sort of get itself down into this minimum where you have a high degree of correlation. And now, of course, if you want to try to learn a new song, you're now kind of stuck in a well that makes it very difficult to get out of, or at least more difficult to get out of, which is why it's harder now for the network to learn a new fluctuation. We added in different kinds of perturbation. I won't actually spend time on that. We not just having neurons drop out, but shift in time or pause and come back again with the same general pattern. So one can think about this perhaps as sort of somewhat analogous to dropout in machine learning. You have a network where you will get a more stable or more generalizable solution if you allow certain nodes to drop out over time. So this is a very maybe simple variation of that idea. So in machine learning, dropout forces the neural network to learn more robust features that are useful in conjunction with many different random subsets of the other neurons. In our case, it just provides us with a network structure that is that's sort of sitting at a more adaptable point. It's able to move to learn new solutions more rapidly. All right, so everything we've talked about so far has been in the framework of this feet and sung model. One issue with that might have occurred to some people is that the biological learning rule that we used wasn't all that super biological. And so many years ago, 2007, postdoc of mine, Michael Faries, who had come from David Pacal's lab, was really bothered by that. And he wanted to be able to train the network, train RA, to generate this sequence of spikes that was very well timed, but using only mechanisms that he felt were within the biological compass. And that is to say, he wanted to be able to do this reinforcement learning problem using a true biological learning rule, which is spike timing dependent plasticity. So many of you will know how that works. What that tells, the way spike timing dependent plasticity works is that one strengthens or weakens the synapse depending on the time difference between the arrival of the presynaptic and the postsynaptic cell spike. And so if you get a cell whose presynaptic neuron fires and then you get a postsynaptic spike within some time window afterward, you would then increase the strength of that synapse. If they occur in the other direction, you would decrease the strength of that synapse. So given some input from HVC driving the RA population, can we, even without this variable input from LMAN, can spike timing dependent plasticity on its own generate a stable series of well-timed spikes at the output? And so answer without modification of the spike timing dependent model rule is that no, not really actually. So if you want your RA neuron to fire at a particular time, we implemented both an additive STDP rule and a multiplicative one. And they both kind of go wrong in different ways. So the additive one the spikes tend to process forward in time. In the multiplicative one, you tend to get more and more spikes over time. And so neither of them just out of the box actually work to solve the problem. So the solution that Michael proposed at that time was to not just allow spike timing dependent plasticity to just run wild. What we need to do is to control it by modulating the degree of spike timing dependent plasticity that we allow in time according to feedback, right? So this is what makes it reinforcement learning. We would like to be able to use the quality of the output to modulate the degree of plasticity in the HVC to RA connection. And so he defined a reward as a function of time, which was the difference between the output spike train and the target spike train, and used that as a temporal modulation of the strength of the learning rule. So this was an early, I'd say, use of what has now kind of entered the literature quite a lot more strongly as a sort of a tripartite learning rule that doesn't just depend on the presynaptic and the postsynaptic spike, but it also depends on the third factor. And in this case, that third factor was some kind of signal that let the system know about the quality of the output that would be generated from the spike, from the output of the network on that trial. And so he showed that you could use this to learn more complex patterns, and that in general you could get decent performance. So if you used general Poisson input, that is to say your HVC neurons were allowed to have kind of a Poisson firing train that was driving the downstream RA output, then you would get okay performance if you wanted every one of your RA neurons to fire only once. If you were demanding that they would fire multiple times, then it gets hard for the system to figure out how to assign the credit appropriately, so the performance does drop off. What makes it a lot better is if your HVC neurons only fire once, right? So if you make your HVC neuron very sparse, as we actually see in the real system, then the performance is much higher. And that is, of course, because the system is able to kind of use time as a kind of cue for credit assignment, that it's only the neuron that's firing at that particular time that needs to care about the imposition of that error, which you're not allowed to just direct specifically to a particular synaptic connection. You've got to kind of give it to everybody. All right, so that, you know, was an intriguing and novel suggestion, but I think some of you will have noticed a problem between our original circuit diagram, such as it is, of the bird brain and how both of these models are working, right? So we've introduced a model that kind of gets around some of the non-biological realism of the original model by thinking about that modulation as being kind of a global signal that modulates in time. However, you know, that green, right, the modulation in the case of our model, and the critic in the case of the feet and Sung model, is being given directly to RA. Now, you can see from this picture that there is no critic, right? So we would think about that typically as, say, being a dopaminergic input coming from VTA. That input does not reach RA. RA has pretty much no dopamine coming into it. And so what is the mechanism for creating that error signal, and how does it reach the synapses that we need it to reach, which are those between HVC and RA? So one hypothesis is that the entire anterior forebrain pathway kind of acts as that critic, right? That the VTA is a component. It's you know, it is able to encode errors, but something about this entire forebrain pathway is able to act as kind of the evaluator and the critic, and potentially to play the role of doing that modulation in plasticity that we would need it to do. So why is that plausible? We argued in this paper that there are a couple of reasons why that might be reasonable to think about. For one, as I mentioned before, that the L-man synapses into RA are NMDA, which is nice, right? It means that they are well placed to generate NMDA dendritic spikes, and thus to influence HVC to RA plasticity. There's another kind of neat aspect that L-man is able to fire in two different modes. It can fire both in where the spikes are kind of isolated, just single spikes, but it can also fire in a different mode where it becomes bursty. And potentially bursts could be a way that would heighten the effect of STDP. So there's still a problem, right, in that we would like the error signal to arrive at the same time as the incoming information from HVC. If we wanted to tighten up the synapse from HVC to RA, we need to know sort of simultaneously that that is a good or a bad connection to be operating. And so that kind of requires that the anterior forebrain pathway can predict the output. And that also has some degree of plausibility because HVC does in fact also project into area X. And so you could think about that as sort of a motor efference copy, that if the anterior forebrain pathway kind of learned to act kind of as a forward model, it could predict how that output would drive RA and generate a version of the error. So that might be a little bit too strong. And so there have been some recent models that have sort of taken up that idea and thought about the possibility that the anterior forebrain pathway is kind of acting as a tutor effectively, that there can be somewhat more rapid learning that is taking place, partly via the input from VTA that trains the anterior forebrain pathway to make a prediction, and that then helps to tutor the HVC to RA circuitry. And so there's models of that type by Jesse Goldberger, Michael Fee, Girey et al., and a recent very nice paper from Bensi Olvetsky's lab. There is good experimental evidence for the existence of such a situation where you have sort of two-stage learning, both in the anterior forebrain pathway and in the HVC to RA projection. And that comes from this. So this is the same experiment that you saw before, where in the same paper, in fact, where Michael Fee's lab were training the bird to shift syllables. And then what they did, so they would train it in the way that you're seeing on the screen, and then they would block output from LMAN. And what they found is that the pitch that the bird would sing at would go back to the one it had learned the day before, right? So as though any learning that was being done in the HVC to RA channel was being sort of augmented by extra bias that was coming from LMAN. And if you get rid of that bias, then what you sort of reveal the state that the HVC to RA pathway was at sort of the day before. So this strongly suggests that there is a kind of bias being learned within the anterior forebrain pathway. We can't say rather whether it's in LMAN or the rest of the circuit, because LMAN is the only output of that entire circuit. But there's a bias somewhere in that pathway that is contributing to today's learning that will be kind of reinforced overnight, perhaps through sleep or whatever mechanism. All right, so let's go and look in a bit more detail at what the anterior forebrain pathway might actually be doing. So in the picture that I talked about with Michael Ferry's paper, we thought about the anterior forebrain pathway is basically they're providing the modulation signal for plasticity. That sort of took away, sort of changed the focus of what that pathway might be doing from the original model from Ila and from Terry Sinowski, that the anterior forebrain pathway is all about injecting variability. It certainly does inject variability, so we know that's happening. So maybe it's some kind of hybrid between the two things. So one way of thinking about it is maybe that LMAN itself is basically it does act something like a random number generator, but it's real biology, so we'd like to know how it does that. Maybe it acts like a chaotic network, right? One might think about it as sort of a generic cortical circuit that has both excitatory and inhibitory neurons. If they are in a balanced state, then the natural mode that they would operate in is to generate chaotic outputs. Then area X, one could postulate, acts to compute the signals that are needed to modulate the LMAN output. And that can be trained or modified by signals coming from VTA. So LMAN, variability generator, X is the signal that kind of moves around or controls chaos coming out of LMAN. And that, the idea for that, or, one beautiful line of work that sort of shows how that could work comes from Guillaume Lejoie and his work with Eric Shea Brown, where he showed that, while we think about a generic EI network as generating chaotic output, if you in fact give it a time, a time varying input, you can control the amount of chaos quite a bit, right? So you can sort of dial the chaos up and down by the degree to which you drive the network, either by just changing its DC level, where you can just make it more or less chaotic, but you can also control it very tightly in time by potentially driving the network strongly by external inputs that kind of lock it into a much lower dimensional chaotic state. And just to show that Area X can have a role like that, here's an example where we've sort of have some ideas about how that works from the ground up. So Area X, we know, drives contextual variations in adult song. So everything we've talked about before, we've been thinking about the role of these inputs as sort of modifying plasticity on perhaps very rapid timescales. We know that there is a kind of slow timescale modification that the activity coming out of Area X can do, and that is to change the bird's state from the case in which it's singing alone, which is called undirected song, to when it's singing in the presence of a partner. And so that's somewhat like your songs where you're singing in the shower versus your kind of performance mode when you're singing in front of your crowd that you hope to impress. And the difference, there is a difference in the nature of the song. So during undirected song, if we just look again at the frequency of that well-defined harmonic stack, that is quite a broad distribution in undirected song, where it becomes very narrow in directed song. And so there is something that is altering the intrinsic variability that is being injected into the song. If you look at LMAN, right, so the output of the anterior forebrain pathway that's injecting input into RA, you can see what's going on. So you look at the undirected case and you see that the neurons are firing many times throughout the song, whereas during a directed trial, and again here, we should point out that difference in the firing properties, right? In undirected song, there's lots of bursts and they're scattered all over the place, whereas in directed song, you just get single spikes and they're considerably more locked to timing in the song. So what is Area X doing? That's what the output from the entire pathway looks like. We need to then look in a little bit to the internal structure of Area X. So Area X has two primary important cell types, the spiny neurons, which get the inputs from HVC, and the output neurons, which are the palatal cells. So those of you who know something about basal ganglia, these are neuron types that you also see in basal ganglia. Palatal cells fire continuously in this kind of tonically firing way, whereas spiny neurons are just driven by, they're excitable, just driven by punctate inputs. So the spiny neurons, the input neurons, show very little variability, very little change between contexts. The directed song and the undirected song, they fire in pretty much the same way, reflecting the nature of the inputs from HVC. Whereas the outputs from Area X show strong modulation by context, right? The directed song and the undirected song, they have much higher firing rates during undirected song, and you get these big pauses in the firing that are very variable in time. Right, so we know that Area X is doing something different in the two contexts, and that we believe is because it receives a lot of dopaminergic input. So dopamine is, the application of dopamine is able to mimic the kinds of changes that you see in context. So here again, you're just looking at a distribution of the fundamental frequency of the particular syllable in the song. When the bird is singing alone, right, it has this broader distribution, and when it's singing in the presence of the female, it has a narrower distribution. If now you add an antagonist to dopamine while the bird is singing, then you make the bird, you recover the variability that's observed in the bird as if the female weren't there. And so it looks like dopamine is kind of the switch that moves it between these two modes of singing. In an extensive project that I won't tell you all the details of, we kind of chased down a new neuron type that David Packel and his lab observed in Area X, which had some very interesting properties. It was regularly firing, and it seemed to drive the output palatal cells very strongly. We also found that that an output from that cell would sometimes drive a positive and a negative fluctuation, and sometimes would only drive a positive fluctuation. And that the balance between the positive and negative and the positive only was toggled by the presence of dopamine. What we were able to show is that that little circuit motif was able to generate changes in the output palatal cells that can really change the nature of the population code coming out of area X. So without dopamine, you get the circuit motif that looks like this. And two palatal cells, because of, I won't go into all the details, but sort of chaotic dynamics of that microcircuit, those two cells will fire asynchronously and very variably. Whereas when you have the alternative circuit here, where you get the positive and negative inputs that are very tightly coupled, all the output neurons end up firing together in phase. And so we hypothesized that this cell type might provide a dynamical mechanism that allows dopamine to be able to move the output of area X between highly variable and uncorrelated to being very stereotyped, very regular, and also very correlated. So here is an example where single neuron and microcircuit dynamics can shape the nature of the population output. All right, so we've talked about what could be going on in area X. We'd like to be able to predict how that generates structured output in LMAN. And so the next step that we've been working on is to try to build a chaotic network to mimic what's going on. It won't necessarily be chaotic, but to build a network that mimics LMAN, where we've been wiring up the neurons in a geometry that is equivalent to the real nucleus. So something to know about this is that there are really a countable number of neurons. It's something like 400 neurons, and they're in kind of a ball. And so we really did want to understand how this small numbers and the geometry of the network matter. And if you look at the EM data that's coming out of this circuit, Winfried Denk has been very interested in doing some of the looking at the fine structure of some of these nuclei in the bird brain. You see that the typical range of a dendritic arbor in this nucleus is about 200 microns, and the whole nucleus is about 1 millimeter across. And so the extent of coupling among different neurons is something that we'd like to model and like to understand. So we've run different variants of the model with different arborizations, and hopefully, we'll be able to see that the dynamics that one gets from these models, we'd like to be able to match that up with emerging data that's coming out of Michael Fee's lab. So here's just an example of the model running, where now we've given the range of arborization quite a long radius, so about half the size of the network, and you can see that the activity is distributed all around the network. As one reduces that radius, you start to get kind of traveling waves that move around the outside of the network. So we have been looking at trying to match some of the experimental observations. So in this case, looking at the correlation between neurons as a function of their separation, and trying to see what are the ranges of sort of input biases and weights that are consistent with the data that one gets from the experiments. And so the data that we've seen from Galen Lynch's thesis work with Michael Fee, they see something like this, where you don't see a strong dependence, in fact, on distance, which is what you might have thought would happen if you had neurons that were primarily coupled with other neurons within 200 microns. Instead, you find that it's almost distance independent, but there are occasionally neurons that are extremely correlated with one another. So we have been exploring our network parameters to see what range of parameters are in fact consistent with that data. The other thing we'd like to be able to reproduce is the bursting nature of the firing patterns. These networks in general don't tend to burst intrinsically. And one important question that was probably on some of your minds, we've been always, in everything I've been talking about, sort of ignoring DLM, what's going on in DLM, right? Anything coming out of area X has to pass through this thalamic area, and that has very particular form, right? Thalamic neurons tend to do this kind of burst behavior where they can get, they do sort of post-inhibitory rebound and generate bursts, so that, people who've worked at all with LGN know about that kind of behavior. Area X, the palatal cells act to inhibit DLM, so there's a kind of sign change between the coding coming out of Area X and that the DLM will transmit into LMAN. Fortunately, Michael Fee's lab, again in collaboration with Michael Faries, have built a really beautiful model that very accurately predicts the output from thalamic neurons as a function of the ISI the pattern of interspike intervals coming out of Area X. So we use that model to predict what spike trains out of DLM should look like. And you can see right away that you expect there to be bursts that are driving DLM. And so it's very possible that when you get bursts in LMAN, those are actually being driven directly by the outputs of DLM. All right, so those are still rather preliminary projects. And what I hope you take from them is that, there's a lot of complexity to this system, and it all kind of rests on a lot of knowledge of the underlying biophysics and circuitry of the different regions of the circuit. So there's a lot of open questions. We've really focused here on sort of realistic learning rules between HVC and RA, and I've told you a little bit about chaotic dynamics in LMAN. One might also imagine that, I find it very appealing to think that LMAN is not just a black box. It doesn't just generate any fluctuation, but maybe over development, it learns to generate a lower dimensional space of fluctuations that maybe allow learning to happen more rapidly, right? So it's not just purely random, but it's kind of directed or a constrained space of fluctuations that are kind of within the space of reasonable for birdsong. And so we're interested to try to understand how potentially LMAN could be trained to be kind of a low dimensional variability generator that's matched to the natural statistics of song. We talked a bit here about transformation through the thalamic nucleus and how that can depend on the detailed biophysics of the DLM neurons. The detailed biophysics of Area X is going to constrain what kinds of computations it can do, how it interacts with inputs coming out of VTA. We have another project I mentioned earlier of trying to analyze in detail the dopamine signals that are actually generated during song, and we have a very nice paper in preparation about that in collaboration with Vikram Gadekar and Jesse. And finally, how does HVC create and robustly maintain the timing scaffold of the song? That's another fascinating problem with lots of implications to it that I don't have time to talk about, but hopefully next time I get to talk to you, we'll be able to tell me more about that. All right, so I hope I didn't go over, wasn't able to really keep time, I'm sure I did, but here's all the people that have been involved, my collaborating labs and my funding sources, and I'm going to unshare my screen and see you and take questions. Hi, Pravish, I can see you now.
Okay. Finally,
finally we can talk. It's been lonely out here.
Thank you very much, Dr. Fairhall, for the presentation. And apologies for the problems in the beginning.
That was complicated. Sorry about that.
I had to introduce you, but I had everything ready. But I think you really proved that you are truly somebody who needs no introduction.
Thank you. All right, so I'd love to take questions.
Yes, so there is a question on the questions tab. Are there comparable hypotheses in human motor learning about instantiations of reinforcement learning modules and algorithms?
Right, there are. So the person to look up is Benzi Olvetsky. So I mentioned his, he used to work for Michael Fee and has done a lot of beautiful work in birdsong, but has branched out into working with rodents. And so a lot of the ideas that he's developed in the context of the birdsong circuit, he's kind of porting over to think about how learning works in rodents. It's more complicated, obviously, because, I mean, as I kind of stressed at the beginning, the beauty of the birdsong circuit is it only is learning to sing a song, whereas in general motor learning, you're trying to learn many things. And so Bensky's been sort of training his rodents to learn very complicated yet fixed tasks. And so it's a really nice paradigm. Michael Fee has also published a very nice paper about oculomotor learning, where some of the ideas that he has proposed about this kind of two-stage learning, he has thought about how they could apply to reinforcement learning in the oculomotor system.
Awesome. And I had a question from my side. Do you see such approaches? I know that it is the case with the bird songs. Do you think that this approach could work well across other microcircuits in the brain, like with this framework of reinforcement learning?
I hope so, right? Because I mean, Konrad and I could differ on this, but backpropagation, there may be ways to do it, but it does not strike me as the most obvious way for biological systems to learn. I do think we have more legs to stand on to think about how reinforcement learning, or sort of a hybrid between them, right? I think the model that I talked about, the one with Michael Farry's, is sort of a bit of a hybrid where you kind of tell neurons when to change, even if you're not exactly instructing them with a gradient. And so I think there are ways to interpret reinforcement learning that are kind of coming closer toward a kind of gradient-based, almost backpropagation-like strategy. And I do think that that's probably a very general thing.
Yeah, that's interesting. I think that would pave way for more investigation and research in that direction.
I'm certainly hoping to see that. We also have a project with Vanessa Ruto where we're looking at the role of dopamine in the mushroom bodies in Drosophila, and I think that could be another place where and there's actually some very nice work on the archive by Ashwin, by Ashok Kumar, who has done a little kind of RNN model of learning strategy in the mushroom bodies, where he's able to explain some of the weird effects that we see in dopamine. So I think that there are a few converging lines of thought about sort of thinking more broadly about how dopamine's working and what role it might be playing in general in learning models.
Yeah.
So a question here, are we thinking about the role of androgens to encourage the growth of song? Not at the moment, although that does bring up a cool project that I'm hoping I will get to collaborate with Tim Gardner. So I don't know how many of you are birdsong people, but many years ago, Tim did this amazingly beautiful work where he trained canaries when they were babies to sing kind of a random set of syllables. So canaries learn incredibly well, and they were able to produce this sort of almost white noise sequence of tones. When they matured, when they kind of went through puberty, that song that they had learned kind of crystallized itself into a song that had a lot of the structure that I've been trained with, but sort of reorganized into the general pattern of a canary song, right? So the syllables kind of broke up to be canary-like. And so I do think that there's a really important sort of, nature nurture thing going on where there's a lot of learning that's just sort of, trial and error from sensory feedback, but then deeper structures that are triggered by things like hormones. And so that is something that I'm really hoping to interact with him about in the future. He's very interested in getting back to canaries in particular and looking at development and how sort of species-specific grammar might be implemented by the action of hormones.
Great. I think there's one last question.
So it wasn't exactly that, right? So what, it's a little bit of a complicated story that I definitely didn't go into details on. So where we, we used dopamine, so dopamine-ish signals in a couple of different ways. One, we used a signal like dopamine, or like what dopamine might be thought to do, to kind of modulate the degree of synaptic plasticity in our model. We also have a theory, which may not be true, but is a nice idea, that what dopamine is doing in area X is to affect the coupling between this neuron that we saw and the output palatal neurons. Dopamine is a complicated molecule, and it has many effects on excitability and on plasticity. And so the particular effect that we modeled with respect to area X was that it changed the ratio of purely positive to coupled positive and negative outputs. So maybe it's, there are some neurons that have been shown to be both GABAergic and glutamatergic. They're able to give a positive and a negative output. And it could be that dopamine is selectively kind of modulating one of those relative to the other. And what that led to was a different dynamics of the circuit that changed the nature of the firing from being kind of regular to being irregular. So it's a rather complicated role for dopamine, but but one which I think is, again, coming back to the discussion that we had during in our panel. Biology can do lots of interesting things if you take into account the underlying biophysics, because there's a rich slew of dynamics that it can modulate and change.
Okay. So with that, thank you very much, Dr. Fairhall.
You're very welcome. Thank you so much for having me, and sorry to hold you up at the beginning there.
I think finally it worked, and we actually did a demonstration of lifelong learning before your slide. So I think it fell in place, and thanks to Guillaume for jumping in and helping.
Thank you. I should have got another phone sooner. All right, see you guys.
Yeah, thank you.
Bye.
Okay, I hope I'm visible now to all of you. Okay, perfect. Okay, with all that, we have the breakout session with our mentors, wonderful mentors who are setting things up. So we will shortly give you instructions about accessing the mentor sessions. Before that, I think we can have like a pre-closing session with the most important thing, thanking everybody who made this conference what it is. So to start with, first, a very, very big thanks to all our speakers and mentors. A Saturday, even during these lockdown times, it's not an easy thing. So people really believe that this community of neuroscience and AI would be successful in coming together on a Saturday and discussing and have productive conversations. So lots and lots of thanks to all our speakers and mentors, and to all the participants who tuned in from different parts of the world and who stayed since the morning. And I hope all of you have some takeaway from all these. And further, I'd like to thank all the organizers from my colleagues from the Unique Student Symposium organizing team, Stephanie, Surya, Naz, Bertha, and Dongyan. And we extend our thanks also to the unique student committee members, Jessica, Elizabeth, and Raymond and Annalise, and of course to our wonderful Dr. Karim Jerby. So, and to all the partner institutions of UNI and the Centre de Recherche du Québec, which is funding UNI. So, thanks a lot, and I applaud on behalf of everybody to everybody who made this possible. So now about the breakout sessions. We will shortly be seeing a link with, so to all the registered participants, there is a link with all the resources that was shared to you, a spreadsheet, a Google spreadsheet. So I am not sure if somebody posted it here, but I'm posting it again for your reference. This has the URL to all the breakout sessions, the topics, and the mentors in part. There's also a link to the Slido with questions and polls relevant to the topic. So the idea, the modified idea right now is to actually have two breakout sessions. There will be 11 breakout sessions in parallel till 6 p.m. And the idea is to give an opportunity to each participant to attend 2 breakout sessions, unlike the 3 breakout sessions with the original plan. But I guess you would understand the time. So the first breakout session would start in about 5 minutes, and at the end of 30 minutes of the first session that you are a part of, there will be a timeout, and people will be asked to join their session. And so in that way, each participant gets to attend 2 different sessions. So that's the idea. I hope it's clear. You can refer to this document for any information about the mentor sessions. The mentor sessions would be on Zoom. It's recommended to use the Zoom app. If not, it also works on the browser. So I think we can get started. So let's call an end to the crowdcast, and let's all connect on the respective mentor sessions. I will keep the chat on this crowdcast active for about 3 to 4 minutes for people to get hold of the link that I shared right now with all the mentor URLs. So have a good weekend. Stay safe, stay healthy. Let's hope for a better collaboration with Unique in the future. See you guys. Bye.